the press, from the press and other sources, that there's this thing called the gut microbiome. And that gut microbiome we're seeing an increasingly play an important role in various human diseases. And the gut microbiome constitutes basically all the sort of bacteria that live within our gastrointestinal tract. And as I said, increasingly we've seen that there's a sort of crosstalk between the gut microbiome, the immune system, and that can affect plenty of diseases, and that can include things like affecting development of cancers, neurodegenerative diseases, heart diseases, metabolic diseases such as diabetes, and even diseases of the skin. Oops. I go back. And in fact, if we look at the proportion of cells that are human versus microbial, we're actually more foreign than human. So we're actually composed of 39 trillion microbial cells, whereas only 30 trillion human cells. And we're far outweighed by microbial genes. There are 2 million microbial genes living within us, but only 23,000 human genes. And actually, when you look at this data, just a simple piece of data, you're really quite taken aback that how important the microbes, not just within our gastrointestinal tract in our gut, but also on our skin surfaces and other mucosal surfaces play an important role. And it was actually this biologist called Elie Metchnikoff who actually looked at chronic systemic diseases, and he thought that actually, this is back in the sort of 19th century, he thought that there was a relationship between the gut in terms of how well the gut lining is intact or permeable, the microbiota and the immune system. And he thought that actually many chronic diseases are a result of bacteria going across this barrier, which should be intact in health, and into peripheral organs. And we've seen that there is in fact an interesting relationship between how well this lining of the gut is intact, the type of bacteria, the intestinal microbiota, lie inside the gut, and oops, I can't see that one, and diet. And diet can play an important role in shaping how the gut microbiota are in terms of their constituents, because there's these trillions of bacteria within the gut, and then how that can affect in terms of some of these bacteria cross this gut barrier and into the systemic circulation, or if indeed affect the type of immune cells that we all have. So what I thought when I started out this project about 4 and a bit years ago is there a gut microbiota immune axis? Is there this crosstalk between what's in our gut and our immune system in health and versus chronic disease? And we know that the gut bacteria are important to break down certain carbohydrates. In fact, we wouldn't be able to absorb carbohydrates if we didn't have these gut bacteria. And I've mentioned just 3 of them here: Bifidobacteria, Clostridium, and Bacteroides. And these produce these substances called metabolites, which I've listed some of them here, these polysaccharide A or hydroxyindoleacetic acid. And these metabolites, we have seen, can affect the type of immune cells that there are. So whether there are certain immune cells called B regulatory cells or T regulatory cells, which act as a balance to effector lymphocytes. So in human normal physiology, we have some immune cells which are called effector lymphocytes, which are useful to get rid of things like viruses and bacteria, but can be damaging to an organ transplant. And on the other hand, we have regulatory lymphocytes, and these acts as sort of brakes on the immune system and prevent an immune system from going into overdrive and potentially causing things like autoimmune disease such as type 1 diabetes, but also actually keeping in check any potential immune injury to an organ transplant. And what we think that happens in patients who have chronic kidney disease, that the gut barrier, instead of this being very tight, it becomes more permeable. That drives a decrease in the type of bacteria. So instead of having different types of bacteria, we have lower microbial diversity. And we've seen that in patients with chronic kidney disease. This changes the gut barrier function. And then we see these mediators that upregulate effector lymphocytes. So you have more immune cells that can be damaging to a transplant, and a lowering of these regulatory lymphocytes. And this was sort of the thought process that we had, but we've obviously got to prove it. So our hypothesis in kidney transplantation is that we think that in patients who develop rejection, they have a loss of gut barrier function, and that drives a change in the type of bacteria that's lining or inside the gut. And this can then lead to changes in these metabolites, which can tip the balance into more higher proportions of effector lymphocytes, so cells which are immune cells which can be injurious to an organ transplant, and a lower proportion of regulatory lymphocytes. And those are lymphocytes that can be protective for a transplant, so limiting the immune response to a transplant. So our aims were firstly to assess the gut barrier function in patients undergoing kidney transplantation, characterize the type of microbiota and metabolites, profile these regulatory cells, which are really important to keep rejection in check, and determine if there are actually any associations between some of the changes in the metabolites we see and these regulatory B cell populations in particular. So I just summarized this slide. It's a busy slide, but it just shows you what this involved. So we approached patients undergoing kidney transplantation at Royal Free Hospital. There were about 100 recipients that we aimed to enroll, and 30 live donors. And those live donors were going to act as sort of a control group, because we know these patients were going to be healthy, as sort of the healthiest you can get. And so whether we saw changes in the gut microbiota in terms of transplantation, we could then match to sort of healthy patients. And then at 5 day, at week 4, at 3 months, 6 months, and 12 months, we collected blood, urine, and stool samples from both donor and recipient patients, and then followed up patients to look at whether they developed rejection over that first year of their transplant. And this involved a lot of coordination, but also actually a big thanks to the patient cohort, who are sort of meticulous in collecting their urine and stool samples and bringing it either to them and with them, or us going collecting them from the patient's home, or bringing it with them when they had a clinic appointment. And as I say, none of this project could be possible without the generosity of the patients in our department. So as I said, we took blood, urine, and stool samples at the time of transplant, then at month 1, month 3, month 6, and month 12. We then monitor patients if they developed an acute rejection episode. We profiled their gut microbiota, this very sophisticated gene sequencing technique. We then looked at the immune cells within their peripheral blood using this technique called flow cytometry, where we could actually interrogate the type of cells, immune cells, that were circulating in our transplant recipients. And then we also looked at whether their gut lining was permeable or not via this blood test to measure of this protein called intestinal fatty acid binding protein. And then we also looked at the gut-derived metabolites, the microbiota that were producing those metabolites. Well, we looked at their stool samples to see actually what were those metabolites. So this was a cohort of patients. Actually, we had 93 patients in our transplant cohort and 23 live donors, roughly similar age in terms of mid-50s, predominantly in terms of male percentage, 34% were male versus 47% male in the live donors. A fairly quite a disparate ethnic proportion. So only half were actually from a Caucasian population, Caucasian background, and the rest of the patients, I shouldn't show it here, but from different Black and minority ethnic backgrounds. 30% of patients had a live donor transplant, and 70% had a deceased donor transplant. And in terms of rejection episodes, so about 16%, 13 out of that 93 patients actually got a rejection episode, which we split up into these different subcategories of antibody-mediated or T-cell mediated. So we had a good number of patients, and therefore to actually study, a good proportion of patients to study who actually got a rejection episode. So the first thing we did was to assess the gut barrier function, and we did this by measuring this blood level of this protein called intestinal fatty acid binding protein. This protein is usually released from gut epithelium, the cells lining the gut barrier. And we actually saw, if we looked at patients who did not develop rejection, those were blue, versus those who did develop rejection in red, our live donors in control, we saw that at the time of transplantation, those patients who then went on to reject their kidney actually had a higher level of this fatty acid binding protein in their blood compared to patients who didn't develop rejection. And this was really quite an astounding finding, that this protein was higher in these patients that went on to develop rejection. So this just showing that there's probably a more sort of leaky gut in patients who go on to develop rejection after their kidney transplant. We then looked at the different type of bacteria. This is a very busy slide, so I apologize. Compared to patients with no rejection and rejection, these are the different type, all the different classes of bacteria. And what we actually saw, there's similar profiles in patients at baseline who go on to develop rejection versus those who have what we call stable graft function. And this diversity for both actually decreases after a transplant, and we think that's probably because of the immunosuppression having effect on the type of bacteria that colonize our gut, and then it recovers over time as the amount of immunosuppression that we give patients is less intense. And we saw that actually after transplant, compared to our live donors, who are sort of very healthy, don't have any kidney disease, not on immunosuppression, we saw that the diversity of the gut microbiota does decrease compared to healthy controls, and that shows you sort of the sort of robustness of the that's expected, and it just confirms the robustness of our approach. So what about the metabolites? One of the metabolites that I want to just mention here is tryptophan. Tryptophan is present in things like eggs, dairy products, poultry, meat, and in normal homeostasis, in normal health, there are metabolites that tryptophan is broken down by the gut microbiota into things like indole-3 acetic acid, 5-hydroxy indole-acetic acid, and these are important because they actually control this population of B-regs, these B-regulatory cells that are important to control and dampen down immune responses. And we saw that if we measure the tryptophan in the stool, we saw that after transplantation, for both patients who reject and don't reject, that level increases, that tryptophan level increases, and that's probably because now that patients have had their transplant, they are on a sort of unrestricted diet and they can increase their tryptophan level. However, even though both sets of patients increase their tryptophan, the metabolites that they produce, the indole-3 acetic acid and 5-hydroxy indole-acetic acid, this is sort of downstream products of tryptophan, we saw that those were reduced in patients who reject. This is much lower in the red for both these two metabolites. And this is usually at the time point at where the actual rejection point happens. So what about this immune population, this regulatory B cell population that is really important in controlling immune responses? What we saw, again, at the time point, just at the time of transplantation, so this is so when the patient's entering a transplant, we saw that patients who go on to reject, this red group, had lower levels of this important immune regulatory cell subset. This cell subset, as I say, is acting as a brake on the immune system. And then this is also true at 6 months and at 12 months. So quite clearly showing that in patients who reject, they have the low level of this regulatory cell, and therefore that explains probably why they are rejecting, because their effector immune, that balance that you saw, is more tipped towards the effector population and lower amounts of regulatory cells. And then could we correlate these two things together? Could we correlate the levels of these metabolites with the regulatory cells? And yes, we could. We could see that patients who have low levels of this, I just highlight here, the low levels of these regulatory B cells have lower levels of indole-acetic acid, one of the metabolites that I showed earlier that was lowered in patients with rejection. So there is this sort of interesting correlation that we see. And if we put all this together, well, this is the picture that we sort of have developed. We know that the bacteria in the gut, we call commensal bacteria, are important for breaking down fiber and other food substances that contain tryptophan into these short-chain fatty acids. There's 5-indole-acetic acid and 3-indole-acetic acid. And we think that the baseline gut barrier function, these cells, which should be tightly packed together, they're a bit more leaky in patients who develop rejection. And that's true at the point of transplantation, even before their transplant. And that we think that then this transplantation changes the diversity of the microbiota. We saw that compared to our healthy patients, the ones who underwent donation of their kidney, and changes the profile. And that this profile changes to such an extent that this reduces the amount of these immunoregulatory metabolites, these metabolites that come from breakdown of tryptophan, because those patients who reject have lower amounts of these indole-acetic acid, and that's correlated with lower amounts of these B regulatory cells. And because they have lower amounts of these B regulatory cells, that puts those patients at risk of getting a rejection. And we think that restoring these metabolites may restore B-reg function. If we could restore these two important metabolites that control B-reg function, that may restore B-reg differentiation and then limit the development of acute rejection. So we think that there might come the day where we're able to do this for every patient. We can then delineate the type of microbiomes that they have within their stool, determine if they've got low levels of B-regs, and then give them personalized pre or probiotics that could potentially increase the length of a transplanted kidney. As I said, first of all, big thanks to all the participants who took part in this really sort of quite intense study. Lots of people to thank within the Institute of Immunity and Transplantation, where I'm based here, and where a lot of this work was carried out. And a lot of thanks to collaborators at the Institute of Child Health, and to funders from the Royal Free Charity, St. Peter's Trust, the Royal College of Surgeons of England, and National Institute of Health Research at UCL. And this is my group here. A lot of the work was carried out by these two people. Fernando Chang and Amber Vaidkut. And actually, we presented some of this data at the plenary session, which was one of the main sessions at the American Transplant Congress earlier this year. And it was actually, this was voted one of the most impactful pieces of work at that meeting. And just to say that there's lots of other work in my lab. Apart from looking at the gut microbiota, we're looking at developing kidney, miniature kidneys in a dish to model kidney fibrosis. We're developing novel tools to measure certain chemokine proteins in the urine to predict rejection. We're looking at B-cell responses to COVID vaccination in patients with end-stage kidney disease. And this is work we've just started with a new PhD students funded by the Kidney Research UK charities to look at the role of kidney resident cells, immune cells, in controlling two important infections in transplantation, human cytomegalovirus and BK virus. So thank you very much for your attention, and very happy now to take any questions.
Yeah. Thank you very much, Reza. It's been an interesting journey listening to you. There are a lot about transplant we don't know, and you are making us aware. So regarding the medication we take, the anti-rejection drugs we take,
yeah,
how does it affect this microbiota?
Yeah, good question. So we think it does affect it quite profoundly, because we see when we compare it to sort of healthy controls, that is the patients who donate their kidneys who are not on any immunosuppression, we do see this difference. And some of that is going to be because of medication. Some of that will obviously be diet, but we think that the effect of diet is sort of somewhat minimized, because patients who have a kidney transplant tend to have a sort of unrestricted diet compared to being on dialysis. So we do think those medications do have an effect, but it's going to be different for each individual, and that's where the complexity lies.
Thank you very much. Has anyone got any question for Reza?
I can see some hands up from Patricia and Carol.
Carol, go ahead. She was before me. Unmute, Carol.
I was doing that because the football's on next door. Thank you, Reza. It was a very interesting talk. My question actually was the same as Nee's, but could I ask you, is it possible for us to have a copy of the slides to put on the website?
Yeah, I've sent the link to Nee.
Right. Okay.
Yeah.
Thank you.
Thank you.
That was going to be my question as well.
Right.
But I was also going to ask you if you could send us a simplified version, because I understand the study went well, but it was a little bit difficult for me to follow. So
I'm
I wonder
sorry.
if you could send a simplified version.
Yeah, I'm really sorry about that. Is there anything you'd like me to sort of go over that in particular that?
No,
Yeah.
I'll be okay if I get a simplified version.
Sure.
Because
I'll
I'm
send it.
taking the minutes as well, and I need to be
Sure.
able to interpret it for myself so
Sure.
that I can send it out to everybody else.
Okay, no problem. We'll do.
Thank you.
So the leakage you are talking of in the gut,
Yeah.
is it something that happens naturally, or is something that happens when we fall ill?
Yeah, that's a very good question. I think that happens as a result of the chronic kidney disease. The disease actually injured the kidneys in the first place. And I think lots of chronic diseases, not just kidney diseases, but we've seen this in even in things like neurodevelopmental, neurological disorders, and things like Parkinson's, even things like autism, and cardiac disease as well. We've seen that there's these changes in the gut microbiota in patients who have various chronic conditions. Some of it is difficult to tease apart because some of it could be as a result of changes in the diet because of that disease is induced. But whether sort of the chicken and the egg, which one comes first, difficult to know. But we know that if we can potentially restore some of this normal gut barrier function, then that would prove that there is actually a causal link. Because if you could restore that, you can then say, well, actually, if we restored it in some people and they, for example, get lower amounts of rejection, then there's you can prove that. Certainly in sort of animal models of disease, that's been shown, but it's always very difficult transferring stuff from the animal models of disease to actually human disease, because obviously the humans are far more complex. And a lot of my earlier work was in animal models of organ transplant rejection. And actually I saw, we learned lots of interesting immunology, but how much of that could be applied to actually human transplantation, there were limitations. And that's why, this is really important to have work like this, where we can try to tease some of this apart, these complex relationships apart.
Yeah. So we don't have to be scared of, we normally hear of kidneys from drug addicts and blah, 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 blah. That will cause rejection. So we shouldn't be worried about taking kidneys from such people, such group.
I mean, so just to be clear, so rejection can happen whatever kidney you take, whether it's from, what you're describing, an intravenous drug abuser or someone who's lived a very clean and healthy lifestyle. So that rejection can happen because your immune system is, for example, very active, and it wouldn't know whether your donor has come from a drug. So that doesn't affect it. No, it's how your immune system is composed, and what our work is showing is that how the composition of your immune system is very much potentially shaped by the type of gut bacteria that you have. So, and maybe that's a target for a new drug therapy that we could develop that actually restores a healthy gut barrier function and restores, these important immune cells that, as I say, these B regulatory cells that act as a brake on the immune system.
In the absence
would you, sorry.
Hello there.
Go on.
Fumi.
Yes. Would you recommend 1 Yakult a day for gut health?
Sorry, what was
one
that?
Yakult. The picture you show the small Yakult bottles next to the kidney.
Oh yes, yeah. Well, there's I mean, I showed that because that's those are one of the most sort of well-known probiotics, but actually, I think what we need to develop is actually very much patient specific. These are not patient specific. You know, I think what we really to actually have a very effective therapy is actually can we see we can restore someone's gut microbiota function and gut barrier function with a particular tailored probiotic rather than just something you can just get off the shelf. And that's where the sort of challenge lies next.
Hello, yes, I'm Diana, a kidney transplant patient, had transplant since 1985. I'm currently on tacrolimus, mycophenolate, and steroids. So I'm probably messing up and making myself a bit leaky inside. Do I gather if I eat eggs and chicken, fiber, and maybe Yakult or a live yogurt, I can go some way to restoring my intestinal gut barrier?
Yeah, well, the foods which are rich in tryptophan are the ones that you've just described, and we think that those are the ones that are important for at least this part of the immune axis, which is important to keep these regulatory cells sort of more active, as it were, and more frequent to prevent rejection.
Right. So talking about personalization rather than just taking a Yo value or a Yakult.
Yeah.
Will you be asking us on our checkups to bring a stool sample in addition to our blood and urine?
That would be the next phase. That would be a clinical trial we'd want to do next to do that. Yeah.
I look forward to that.
Well, if you could press, if you've got anyone in the National Institute for Health Research to sponsor the trial, that would be very helpful. But that's you know, that's where the vision of where this work is going to go next, and hopefully we have the funding to do that.
How about the Zoe crowd?
Yeah, they'll probably want to take over. They'll probably want to take over the project. So that's
and give us all a nap.
Yeah, but it's something to consider. Yeah.
Right. Thank you.
Reza, could a gut barrier problem be the trigger for vasculitis?
I mean, I haven't looked in particular vasculitis, but certainly there are autoimmune diseases, of which vasculitis is one of them, that we know that there is a connection between the gut microbiota. So things like SLE, we've seen that there's a strong association between SLE and changes in gut barrier function, certainly in animal models, even some in human studies. In fact, one of my colleagues at UCL has studied a lot the effect of the gut microbiota and these B regulatory cells in SLE, and very, very interesting work. So Claudia Maury has done, also based in the Institute of Immunity and Transplantation. In fact, that's one of our collaborators. So yes, I think there is a connection there.
Thank you.
Jill, you've raised your hand as well. Sorry.
Yes, it's okay. It might be the most stupid question of all, but when you mentioned at some point that some people had, was it fatty acids in their gut, which were more likely to make them reject even before the transplant.
So this fatty acid,
is that to do with their diet or?
Yeah. So this fatty acid binding protein, that's not causing the injury. It's just a marker to measure how well the gut barrier is intact. And if you've got high levels of this marker, it's pointing to the fact that your gut barrier might be a bit more leaky than normal. Why that should happen in some people, that's a very interesting question. We know that some people who are on dialysis have more leaky guts than others on dialysis, but we couldn't find the reasons why it was higher. What we did see though was that when this was higher at the point of transplantation, those patients ended up with rejecting their kidney.
Rejection, yes.
Yeah. So it was really a profound finding to see that, and quite unexpected.
But it's nothing to do with really their diet, or is it?
Possibly.
Fatty liver, don't you? Sometimes if you.
Yeah, this is not to do with the actual cause of any fattiness, by the way. This is it's just the name of it. It's possibly diet, but we know that anyway in patients who are needing a kidney transplant, they have a restricted diet, and some might be more restricted than others. Some might be more sort of vegan, and actually those would probably actually have a more diverse microbiome, which is probably related to health than having a very restricted gut barrier, gut microbiota, and thus more leaky gut. Yeah.
Okay, thank you.
Right. Thank you very much, Reza.
Delighted.
For finding the time on such a busy Sunday for you.
Thank you. That was my pleasure.
And yeah, and if I can just ask a quick question. The patients you are using, were they coming from Royal Free?
They're all from Royal Free.
Thank you very much.
Yeah. No, and as I say, a big thanks to all the participants who assiduously collected their urine and stool samples for us. So this work couldn't have been done without them.
Yeah. Thank you very much. I'll get in touch with you.
Great.
As usual, and we'll catch up.
Great. Look forward to that. Thank you very much. Thank you, everyone.
Thank you.
Bye.
Thank you.
Bye-bye.
Yeah.
Bye-bye.
Bye.